Good morning, everybody. Give me a moment. I just got to get rid of something. Do. All right. People will be rolling in. Let's have a look where we're at. God, it feels like Friday already. Don't know about you lot, but I feel like I'm wasted. Partly though, obligatory puppy shot. That's because of this little pest. Yeah, she might look super cute, but she's a terror to this one, who's enjoying a rare moment of tranquility and not being pestered by her little sister. Okay, so what do we got to do today? So today is the last day of class, which is, I don't know, I always get surprised when we get to this point in the semester because it feels like it's, just something that's going to go on forever, but obviously it doesn't. It has to end at some point. And so today are the last two lectures in the last lab, which is, kind of crazy to be honest. And let's get back to where we finished off. And so today we need to finish up control of gene expression in eukaryotes, right? Which is probably going to take at least this lecture and maybe a little bit of the next one. And then we can have a look at some cool stuff, some stuff about RNAi, which is something that I use intensively. It's like a major tool in my toolbox for the research that I do. Some kind of GFP or green fluorescent protein reporter work, and things like CRISPR-Cas, right? So kind of some of the cool applications of what you've been learning, and also as well a way of looking at how we ask the questions that lead us to knowing what we know about genetics, which is also really fun stuff to talk about. So let me get myself situated. I hope Mr. Roberts around. I can't see him. We have not too many with us today, so hopefully anyone that's missing will be able to catch up afterwards. Oh, and as a reminder as well, the exam 4, so that's the last exam, but not a final, right? It's just the last exam. There is no final. Opens tomorrow at I think it's 9 a.m. I'm just going to type this down too. What would that be? 28th. Feels like summer's almost gone, to be honest. Until 11:59 p.m. Same day. Okay, so a little bit different. It's not going to open today. It's going to open tomorrow, and part of the reason for that is that the aim is just to kind of almost enforce a little bit more studying time, right? Because some of the things that we're going to talk about today are fairly advanced, right? So I want you to have time for those to kind of percolate through. And also, if you have any questions, to give you an opportunity to ask me them before the exam, right? Which is also important. Okay, so we finished up on Thursday talking about, finishing up about epigenetics, right? So just to recap, epigenetics consists of kind of 3 main kind of mechanisms, I guess. One is chromatin remodeling, right? Moving histones around or off of DNA. No worries, Marcus. I was just showing everybody my dogs again. Oh, cool beans. Thank you, Dipper. The second mechanism is the modification of histones, right, directly. And really that's all about affecting how tightly histones bind DNA. So the chromatin remodeling is like large scale, like moving things around and off and back and forth and stuff. Histone modifications directly are about affecting its interaction with DNA. And so there are two main mechanisms. Acetylation by HATs, or histone acetyltransferases. If I can spell that. Oh, and also demethylases, demethylation. Equals pro-gene expression. Deacetylation by histone deacetylases, not too surprisingly. Also called HDACs, which you'll come across again in the future, I hope. And methylation by histone methylases. Equals anti-gene expression, I guess, is probably the easiest way of putting it. So that's modifying the histones themselves, like chemically modifying them to have different properties, right? And those properties affect how accessible that DNA is for transcription and therefore controls gene expression. And then the final one is methylation of DNA itself. So as with histones, methylation is anti-gene expression. Have you closed the gate, love? Oh, we'll find out soon. By DNA methyltransferases, or DNMTs. Oh, and that was also anti-gene expression. And so what you tend to find is don't have a good picture of it, unfortunately. You tend to find these motifs, right? These CG motifs, right? So the methylation is of cytosine, right? Which you can see here. It turns it from kind of this shape, size to this, right? And so that basically changes what it looks like, right? In terms of its shape. And so given the proteins recognize DNA sequence based on its shape, therefore things aren't going to bind it as well, particularly transcription factors, right, which are required for recruiting RNA polymerase, turning genes on, turning on transcription, things like that. They also are recognized now by repressors, right? So the whole aim of methylation is to kind of shut down parts of DNA. And it's really crazy because you tend to find these CPG islands, right? These, and the P stands for phosphate. So it's cytosine, phosphate, guanine, phosphate, something else, right? You'll tend to find those in promoter regions, right? And let's write that down. Now, I don't kind of fully understand all of this because it's really stunningly complex. I was trying to read a paper on it the other day about kind of CPG islands and affecting different regions and valleys and stuff. It kind of went over my head a bit, to be honest. But what's really crazy is that quite often in cancer, different types of cancer, obviously, you'll find that the promoters for anti-cancer genes, right? So cell cycle checkpoint genes, things like that, are heavily methylated. So part of like a cancer profile is not just activating growth and development, right, which you need for cells to divide and things like that, but also shutting down the checks and balances that are supposed to stop that from happening. And one of the ways we think, I mean, it's still not fully understood, one of the ways we think that happens is by DNA methylation, particularly of, I guess, what one call guardian genes, right, which their role is to guard against uncontrolled proliferation. So that's kind of an interesting side aspect to this. And also another really cool thing, which is where stuff gets crazy complicated, get you back, is DNA and histones can talk to each other, right? So quite often the DNA methylation of DNA will lead to the methylation of histone to kind of reinforce that shutting down of gene expression, right? So this is like a positive feedback mechanism. And so what you can see here on this slide, let's see if I can make this a bit smaller, is we have a cytosine that's methylated here. This will recruit another protein, whatever MECP2 stands for, methylation of, I don't know what actually, that then recruits a histone deacetylase, an HDAC, right? So it removes the acetyl group, which in turn has an effect of kind of causing DNA to be bound more tightly. And that then also recruits a histone methyltransferase, right, which methylates that histone and so fully shuts down gene expression, right? So this is really kind of, there's a lot of feedback between the histones and the DNA, essentially to kind of have a positive feedback, right? So you don't want to have like methylated DNA, but, you know, acetylated histones, because there'll be kind of odds as to the how much gene expression you're going to get from that region. So it's kind of all one way or all the other, and they talk to each other to do so. I think there's another example here too, which is looking at another mechanism by which that happens. And you see here, so this H3K9, that's essentially, that's the code, right? We know we're talking about the histone code kind of further up in the PowerPoint. The H3K9 basically means that lysine 9 of histone 3 in the octet, you know, in the octamer, actually, it's probably a better word, is methylated. Right, so that's what that code means that you see here. So we have our nucleosome core particle here, and one of the histones in there has one of its lysines methylated, which produces a particular outcome, right? And because there are 8 different histones and they have different lysines, you can see how complex this code can be, right? So it's kind of crazy if you really think about it. You know, we're talking about DNA having like these 4 letters, and those 4 letters encode, you know, essentially amongst them 20 amino acids, and then we get 20,000 different proteins from that. Now, on top of that, all of that complexity, right, how all those are different, controlled, and do different things. Now we have this extra layer of information, right? We have not just the information in the DNA, but in a sense, if you think about it, the information in the histones that the DNA is wrapped around also control whether or not those genes are expressed. Right. So remember ages ago, we talked about how, DNA is just a book, basically, right? It's a store of information. It doesn't do anything. What matters is, when you open that book and what you do with the information in it. Right. And so this is just another way in which you can control when that book is opened and that information is used, which is, I think, it's kind of crazy. And honestly, this is like a whole, rabbit hole which you can dive down and you'll never come out. It is just an astonishingly complex subject, which, kind of goes in one ear and out the other for me, because it's like they're talking a different language, which in a sense they are, right? Talking about the histone code. So all of that information on top of the DNA, that information is, well, all that information on top of the DNA sequence, right? So we've got the DNA methylation, and then we've got the histone modifications on top of that. Much of that, although we don't really understand how, is inherited. So part of what you inherit from your parents isn't just DNA, genomic, mitochondrial, that stuff. It's also those DNA and histone modifications as well. And so you can see how inheritance, we were talking way back at the beginning of the course about, Mendelian inheritance and stuff like that, right? And how, quarter will get this and quarter will get that, kind of working on fairly simple probability. Now you can see that that's really a gross oversimplification of inheritance, because there's so much more going on. It's really quite astonishing. And really, it kind of makes sense when you think about, well, I'm clearly not either of my parents, right? And I'm not a 50-50 mix of the two either. I am bits of them in different ways and me too, right? So it's always interesting, I think, when you think about inheritance, like the more layers of complexity you have on that, obviously the more complex the outcome that you're going to get. Okay, so that's it for the chromatin aspect of gene expression. So now you can see how there's many ways, without even thinking about transcription and the whole central dogma type stuff, in which you can control gene expression, right? Before you even get down to the level of, promoters and RNA polymerase and stuff. Does anybody have any questions about what we've covered in terms of chromatin and histones and methylation, acetylation, stuff like that, before we move on to control of transcription? Sadly, coffee is finished. Don't forget, it's important to know the lingo too. So if I'm talking about DNMTs, I'm talking about DNA methyltransferases, so you're talking about methylation, cytosines, and DNA. I'm talking about HDACs, histone deacetylases, removing acetyl groups. And you need to know the consequences of the actions of those enzymes. HDACs lead to transcriptional repression. HATs, histone acetyltransferases, lead to transcription. So you will be asked questions on those different proteins and the outcome of their actions in terms of gene expression. All right, anyone have questions? Okay, so let's move on to the next kind of layer of the cake, essentially. And so now we get into, again, a whole world of complexity, right? And so our subjects for this, and really I want to kind of tie in transcription, multiple promoters, RNA processing kind of together, because it's all about generating a functional, mature mRNA. So we've got transcriptional regulation, cis-regulatory elements we're going to talk about, and we'll come back and go through these, right? I'm just kind of skipping through them to show you what we're going to do. Alternative promoters, and I've got a really cool example of some work that I did that covers that. Enhancers, which are kind of other regulatory elements, silencers, insulators, and so on. Okay, so that's kind of a whole chunk on transcription, because even though there are many other layers to this, control of transcription is a very key part of control of gene expression, right? Because you need to turn these genes on in the right way and place and in response to the correct signal. Otherwise, it's all moot. Doesn't matter whether your chromatin is open or not if you're not going to turn on gene itself. So in prokaryotes, they're fairly simple in terms of transcriptional regulation. And that's because, well, let me kind of rephrase that. Their promoters are pretty simple. Because generally speaking, if a promoter and operator are open in bacteria, you'll get transcription, right? RNA polymerase will bind, go through, transcribe the gene or the operon. In eukaryotes, it's much more complex because there are two elements to the promoter. You have the core promoter, right, including the Tata box. This is where all of the kind of core transcriptional apparatus will bind. So the Tata box binding protein, the 6 general transcription factors, RNA polymerase, mediator, so on and so forth. Right, so that's for the core promoter. That's kind of like the bare minimum. So again, TBP, binding protein, transcription factors, RNA pol, etc. bind. And that's basically the bare minimum for transcription. However, that core promoter alone isn't going to work super well. So the core promoter, oops, if I can spell it, will give you a low level of basically uncontrolled gene expression or constant gene expression. Extra of there. I didn't see that one, it was hiding off my screen. To be able to get high gene expression or conditional gene expression, right? So gene expression that occurs in the response to particular signals, whatever, now you need the regulatory promoter. And the regulatory promoter is much less well defined. Core promoter is pretty small. We're talking I don't know, like 400, 500 base pairs, give or take. Again, it's not super well defined, but it's smaller. The regulatory promoter can be enormous. It can be quite small. It can be huge, in the range of, I don't know, thousands, if not more, base pairs. It's actually a very hard thing to define. Typically, when you're doing this kind of work, you just amplify the biggest chunk of DNA that you can and stick that in front of whatever you're trying to control, whether it's a fluorescent reporter or whatever. Because there's not really any way of knowing how much DNA you need. It's very difficult to figure out what's part of that regulatory promoter without an awful lot of work. So that regulatory promoter is where things called transcriptional activator proteins bind, right? So and those are what really control gene expression, right? So these, this is called an enhancer, and that's essentially another place in the DNA for these transcriptional activated proteins to bind. There are also proteins that will bind to these sites which inhibit gene expression, typically by blocking the binding of transcriptional activated proteins, right? So there's a lot of kind of interplay going on to generate the outcome of RNA polymerase starting transcription. And so when we dive into this regulatory promoter in more depth, really we start talking about what are called cis-regulatory elements, or CREs. And so these are bits of DNA kind of next to the gene or next to the core promoter, which control gene expression in one way or another, either by promoting it or by repressing it or whatever. So again, a lot of language here. Enhancers are really places which promote or enhance gene expression. Silencers are regions of DNA which bind repressors, and those repressors proteins kind of quite often block the binding of transcriptional activated proteins, sometimes even binding of the core transcriptional apparatus, although that's a little rarer. Insulators are essentially kind of like bookends or walls between different regulatory regions, so you don't accidentally activate genes that shouldn't be turned on. And response elements, these are basically bits of DNA sequence that are recognized by proteins. And so if you remember when we're talking about prokaryotes, they do their coordinated gene expression or coordinated response to stimuli by grouping all of the genes together into one operon, right? So they just have to control the transcription of that operon, and you coordinate expression of all the things that are needed. Now in eukaryotes, we still need to do that, right? If you're making an arm, let's say, there's a bunch of different genes that are required for arm making. You know, kind of making bone, muscle, nerve growth, et cetera, et cetera. However, eukaryotes do this differently. Instead of grouping all the genes together into one transcriptional unit, the operon, what eukaryotes do is have these response elements, which are recognized by a particular protein, in front of all of the genes that need to be expressed in the same time in the same manner, right? So a classic example is heat shock response. You know, if you start heating up a human body, bad things start happening, right? And so there's a coordinated response to that stress. So all of the proteins that help, you know, kind of folding proteins back, you know, stopping proteins from falling apart, all the other jazz that goes on, all have in front of their kind of core promoter response elements that are recognized by a heat shock protein. And so that's how eukaryotes coordinate their gene expression of genes which are important for a particular process. It's not by grouping them together, it's by having kind of tags. It's almost like sticky notes, if you think about it. You know, go through a chapter of a textbook, you put sticky notes for all the things that kind of talk about a particular subject. They have those, you know, sticky notes in front of all the genes that need to be involved in that. So now to turn them on, you just need that protein present, and that protein, wherever it finds that bit of DNA, will bind and activate gene expression. So that's how eukaryotes coordinate their gene expression. Obviously, it's not the same. Not all genes have those, and they will vary. And they will, you can also vary by number, right? So you can kind of control, you can have a graded response to the presence of those proteins. And so those look kind of like this, right? So SV40 actually is it's a virus, which infects eukaryotes. And it's expressed at really, 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 really high levels, right? Because you need like lots of viral particles. So what you see is there's an awful lot of these response elements, whatever the GC boxes do, recognized by a particular protein, I guess. Pardon me. For other genes, you may see some of those present. You may see other boxes. Now, I don't actually know what the CAAT and the OCT box do. You can look them up, right? I keep trying to remember to, and I keep forgetting. Yeah, it doesn't say down there. But essentially, the presence of different boxes controls the expression of those genes in different ways, right? So if you want a lot of expression really quickly, you'll have a lot of a kind of a generic response element, right, which just equals gene expression. If you want to control where, when, or how much expression you have, you're going to have a mix of those response elements. And so these are modular, right? You can actually take one of those sequences out and put them somewhere into front of another gene, and now that gene will be turned on in response to the same or presence of the same transcription factor, right? And so this is actually, it's thought to be the way in which new function can evolve, right, or can appear and then be selected for. So I think butterfly wing spots is a classic example, right? So, you know, they look like eyes, they have the same color pigment as eyes, and it's actually pretty sure that that came about from the response element for eye pigment, right? So making if you're a butterfly, your eye is red, let's say. That response element getting carried in front of a gene that's expressed in the wing, or a response element for, you know, wing expression getting put in front of the pigment gene, right? So now you can gain new function or expression in different places simply by moving those response elements around. Transposons are actually one way in which those things move, which is another interesting point. So this is just talking about one promoter, but you can actually have multiple promoters too. Now I'm going to show you some work that I did a little while ago, right? And so this is a gene called PNC1, which is involved in NAD recycling. So NAD can get broken down into a compound called nicotinamide, which then gets turned back into NAD for other things. Let me just, we'll talk about this stuff later if we have time. So this is what the gene itself looks like. This is the genomic locus for this gene. And you can see it has 5 exons. This is the mutation that that we were working with. Exons 1A, 1B, 2, 3, and 4. You can get 3 different proteins from this one gene. depending on which promoter is being expressed, right? So we have promoter in front of exon 1A, we have a promoter in front of 1B, and we have a promoter in front of exon 2. And so this generates 3 different proteins depending on which promoter is being used. And one of the key points is that if you get expression from this first promoter, this exon has what's called a signal sequence, which results in that protein being exported from the cell. So the full length protein is secreted, right? It's actually kind of floats around in the pseudocelomic fluid of the worm. Expression from the other 2 promoters generates protein which just stays in the cell and does its job there, right? So depending on which promoter is being used, you get a very different outcome in terms of function, right, of the PNC1 protein. And also now you can control, this is a tiny, tiny exon. I don't think it does anything much. But you can control very, very to a fine level where these genes, where this gene is expressed, right? And you can see that down here, and I'll get to what you're looking at in a little bit. Let's just double check, make sure. Yeah. So these are confocal microscope images. You know, I was showing that pretty one of the alligator embryo a little while ago. And this is the head of the worm, right? So this is like the, kind of like turkey basters. These are the bulbs that suck up bacteria and grind them up before going into the intestine. And this is about where the worm's brain is. They do actually have a brain. It's called the nerve ring, about 300 neurons. And when you look at expression of the secreted form, actually you can kind of, let's see if I can get it over here. You can actually see that that protein is kind of floating around in the space inside the worm, right? You know, because it's like kind of flowing around in your intestinal cavity, essentially, you know, equivalent of, right? And these are embryos. You can see the 2-cell embryo here, and there's gone through multiple rounds of cell division, right? So it's doing its function like outside of the cells where it's expressed. But when you look at the other ones, this is the 3rd version. It's expressed largely in the intestine. What it's doing there, we don't know. And the 2nd version, these are all neurons, right? So you can see these are the dendrites, right? Expressed that kind of extend up to the worm's nose. You can see the cell nuclei, right? So these really bright dots. And these are all different individual neurons where this gene is being expressed, right? One version of it. So clearly, this is doing a different job in different places. One version, you know, deals with recycling all over the body, and other versions deal with recycling in very specific parts, right? So all of that just from, here we go, one gene, right? So those different promoters can give you a much kind of broader palette, I guess, in terms of how you use that gene. So I always think that's particularly cool. Okay, before we move on, any questions so far? Anything that you didn't catch, needs going over again? Stuff like that. Okay, so as we mentioned earlier, enhancers are where those extra proteins bind to kind of give an extra boost, right? These are quite often involved in actually activating gene expression, not just recruiting RNA polymerase and stabilizing the core transcription complex. And they can be, figuratively speaking, miles away from the gene which they're helping to regulate. One of my fun examples is, don't know if you've ever seen polydactyly, right, which is essentially where you end up kind of having typically an extra thumb, maybe like that, maybe an extra finger or two, right? Which would be kind of crazy. You can see a lot of them in cats. Let's see if I can actually find a little bit of a tangent. Can't spend too much long. Isn't that crazy? They have like the biggest paws ever. I actually had a friend with a oh, that's cool, Dipper. Actually had a friend back at another institution that had a couple of cats like this, but he never let me take pictures of them for some reason. So anyway, they are just like fabulous at killing stuff, particularly insects, because they have these enormous paws, right? And they look like that. Actually, let's see if we can get some kittens. Kittens. If cats are cute, kittens are always cuter. Isn't that cute? Yeah, it's super weird, but very cute. I imagine they're really good at walking on snow too. That's like having snowshoes on. Um, yeah, I could spend ages looking at kittens. Anyway, that's kind of beside the point. So, getting back to the subject at hand, um, the identity of your digits is in part determined by uh, a protein that's expressed very strongly. Like if you think about having like uh, a limb bud, right, or a hand disc, right, because before it turns into fingers and thumbs, you have a lot of expression. Actually, it's down here. That's right. You have a lot of expression of a protein down here. I think it's called hedgehog. I have to double check. And that specifies you have a lot of it. You have a pinky. You have a little bit of it. You have, whatever that finger's called. Yeah, ring finger. You have a little bit less and so on till you have none, right? And that basically gives you your thumb, right? Or your dew claw if you're a kitten. Oh, that's super cool. Desiree, if you could ever send me a picture of that, that would be fabulous. I'd love to see the monster paws. I'll use that in kind of future. It would be immortalized in genetics class. There you go. Maybe not immortalized, that might be the wrong word. But anyway, essentially you have a thumb here because the expression of the protein here is suppressed, right? And so if you have a mutation in an enhancer for that suppressor, then you get basically you get thumbs down here, right? And that mutation for those Hemingway cats is several million base pairs away from the actual gene itself. Yeah, absolutely. You can get polydactyl humans too. Let's have a look. If I can spell it right, polydactyl humans. Yeah, for example, quite often you tend not to get the extra thumb, which is what's happening with those cats, but you most certainly can get extra fingers. In fact, here's a fun reference for you. Oh, shit, what's that film called? Bruce Willis, Mila Jovovich. Crap, I can't remember what it's called now. Yeah, I'll have to remember another time. But anyway, in that there's a pianist who could only, that particular work could only played because that pianist had extra fingers. There you go. So you can't actually physically play that piece if you don't have those extra fingers. But anyway, so that's all about a mutation in a regulatory element that's many, 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 many base pairs away from the gene that it's actually regulating, right? And so essentially what you have is a kind of change in identity, right, as a result, right? So this region now, either you get changing the wave pattern, which gives you extra fingers, or you have a change in identity, which gives you essentially a thumb and a finger coming out from that side. And that's through the, not only, but in the case of Hemingway cats, it's through the mutation in enhancer, which basically stops that enhancer from working. And so you can kind of think about this also in terms of kind of making this a cell-specific deal, right? And so which genes get turned on in particular cells determines on which activators are present in that cell, right? Because the DNA for all these genes have the same enhancers. Now what matters is whether or not the proteins that recognize them are present, these activator proteins. And so albumin, actually, what does albumin do? I think it breaks down something. I'm not 100% sure what albumin does, to be honest. But crystalline is a really fun gene to talk about because it's what turns your lens cells in your eyes clear. So obviously they need to be free of like cellular junk because otherwise you wouldn't be able to see anything. It'll all be like kind of fuzzy and dark. And so this crystalline gene is involved in clearing out the cell contents from the cells that are in the lens in your eye during development. And so clearly you really only want that expressed in your eyes because, I mean, it's not going to do anything good expressed elsewhere. And so that gene or the enhancers for that particular gene are only going to be present in your lens cells, right? So even though the albumin gene is obviously present in those same cells, it's not going to get expressed because the activators for it aren't present. Absolutely, and actually, Marcus, your lens cells are really weird. I mean, they're very weird. Like essentially, they have a very particular shape. If you ever look at the layout of lens cells, and that's what helps partly focus the light onto your retinas. And they do kind of get replaced extremely slowly, but they don't regenerate very easily. And so that's why kind of like UV damage to your lenses like leads to problems later, glaucoma, for example, or just other issues. I'm not much of an eyeball expert, to be honest. But yeah, they can be replaced, but it does so very, very slowly. They can be easily damaged as well, particularly by UV. So anyway, really cool. However, for albumin, obviously you don't want albumin expressed in your eyeballs, or at least in your lens cells, because then you wouldn't be able to see anything. Albumin being one of the main proteins in egg white, for example. And so you're only going to find the activators for the albumin gene in the liver cell, right? And obviously, those activators are proteins, and so the control of the expression of those activators is also controlled by other proteins. So you get these kind of multiple levels of control, right? And identity, right? Why cells are different, part of that difference is because of these activators that are present. And if, you know, any of you ever get a chance to take my Dev Bio class, you can see how that identity, you know, becomes established over time. It's really interesting. So silencers are kind of the opposite of enhancers, right? They're, instead of being involved in activating transcription, they're recognized by proteins that inhibit transcription, right? Either by competing with transcriptional activated proteins for enhancers, right? So you can have overlaps where you have an enhancer and a silencer kind of overlap in sequence, sorry. So if you have a silencer bound, silencer protein bound to the silencer, now that activator can't bind to the enhancer, for example. And also you can get them so that they interfere with the actual transcriptional apparatus itself, right? And so they overlap with that core promoter. And so this is just one way in which, actually this is kind of in the wrong place. This is really kind of getting back to like this complexity, right, in terms of gene expression, right? So it depends on what proteins are present. And this also then feeds back as well into the chromatin formation too, right? So you can see here, this is basically kind of tadpole form, right, for a thyroid hormone. And that response element is recognized by TR, which is our thyroid hormone, and this other thing, which is a retinoic acid receptor. And depending on what's bound to that thyroid hormone determines whether or not we have transcriptional repression via deacetylation or transcriptional activation through acetylation of histones. You can see like the little tails popping off. And that protein, its presence or absence, this T3 protein, determines the transition between tadpoles and frogs, right? So that's the switch that controls the transition, the metamorphosis process. Okay, so kind of getting towards the end of cis-regulatory elements. The last one really to talk about are insulators, right? And so these enhancers can be many, many, many miles base pairs away from the genes that they're helping regulate. And so you really want to be careful that activators bound to enhancers in one region of the DNA don't accidentally turn on expression of genes that they shouldn't be activating, right? And so insulators are the means by which that is done, right? So essentially they're DNA sequences that are recognized by proteins that can block this process from happening. And part of the way in which it does that is that these enhancers work by the DNA bending to bring that enhancer into contact with the RNA polymerase to activate transcription. And so proteins bound to those insulators basically stop that bending from happening, right? So you can't have something over here bending over and activating transcription because, you know, that insulator protein stops that DNA from bending, right? And so that kind of corrals the enhancer for one gene over there and the enhancer for another gene over there, right? So it kind of stops those from incorrectly interacting. Okay, so how are we doing so far? And again, just saw your comment, Izzy. That's really interesting because polydactyly is much less common in dogs than in cats. Don't really know why. But yeah, it's fairly rare. I've not actually heard of, yeah, I don't think I've even heard of polydactyly in dogs. So if you've got pictures of that, that would also be super cool. Make like a little module of polydactyl pets. Oh, that's a shame. But anyway, Desiree, if you can send me pictures of your cute polydactyl cats, that would be super cool. And if you're willing, obviously, for me to use them in a PowerPoint class material. I don't think there's any changes in. I'll double check. Sorry, I'm not laughing at your comment, Adam. I just read Desiree's. I'll double check. I don't think I've changed anything in this PowerPoint, but it's not really that much of an effort to upload them anyway. But I think they're both still the same. But again, I'll double check. If you wish, just check back kind of this afternoon and yeah, then so not some. Well, I mean, there are three kind of PowerPoints that are really going to be covered in this exam from memory. Just double checking here. Yeah, that's right. So it's really 17 and 18, which are the gene expression in prokaryotes and eukaryotes, and a little bit of, I think, 20. I have to double check, which is more like the kind of transgenics type stuff. But yeah, I don't think I've changed them, but I'll check. I'll probably just upload them again, 17 and 18 at least, after class, just in case. Never hurts, and it doesn't take long. Okay. All right, so that's the control of transcription. Obviously, the control of producing a functional mRNA isn't just that, right? So production of functional mRNA, obviously there's all of that stuff with histones and chromatin and DNA methylation, right? Broad scale stuff controlling whether or not a gene is available to be transcribed. Then we have all of this stuff about cis-regulatory elements and regulatory promoters and enhancers, silencers, and so on, right? That determines whether or not a gene is transcribed. But still, we haven't got to a full functional mRNA yet because we still have two more things left. Let's just kind of, there we go. Type that down. So if you think of it like in equational terms, I guess, you know, it's the sum of, it's supposed to be a 1. Chromatin status, DNA methylation, so on, right? So can a gene be transcribed? Essentially is the question there. Transcription itself, so that's the sum of all of the enhancers, silencers, and so on. Third, RNA processing. And that's in particular splicing. And fourth, mRNA stability. basically how long does the mRNA hang around for to be translated. All four of those things kind of sum up in different ways to result in translation of an mRNA, because by and large, what we care about are proteins. There are obviously many, many other, important untranslated RNAs, but by and large, proteins are what do most of the functions in a cell. And so if we want to know how much protein we're going to get, we need to consider all those different steps and processes, because they all add up, or somehow into that final output. Oh, sorry. I haven't had enough coffee today. So one way in which you control what protein you get is through something called alternative splicing, right? So essentially you will have the whole pre-mRNA produced, but for some proteins, not all of them, but for some proteins, you can control which exons are incorporated into the mature mRNA. So this is this must be for an insulin binding protein, maybe the insulin receptor that's expressed in muscles and the liver, and a few other places. And so now you can control the function of the protein by controlling what exons are included, right? What information is included in that protein. And so if you look at this one, the top one, essentially with all of the exons present, right? So you kind of cut out all of the introns as you would do normally. You produce an insulin receptor which has a fairly low affinity for insulin in the liver, right? The reason for that is that the liver is not the primary site of uptake for glucose, right? The liver stores glucose, but only after the muscle cells have their kind of fill, essentially. So by having a low affinity, it takes more insulin for those liver cells to take up glucose, right? So they only do so after, you've been eating lots of Twinkies basically and not using the glucose up that comes from it. On the flip side, muscle cells, you need a lot of glucose because that's what generates the ATP that allows them to function. So the muscle cells are the main sites of glucose uptake. And so you can change, again, same information, right, in the insulin receptor. But you can change how that protein works by leaving out exon 11 from the mature mRNA. And so that alternative splicing, instead of taking out, this intron and this intron, now you jump from the end of exon 10 to the beginning of exon 12. And so the resulting protein that you get from this different mRNA, still transcribed from the same gene, but a different mRNA, now that has a high affinity for insulin. So whenever even a little bit of insulin is produced, muscle cells will take up that glucose and use it for, making ATP and contracting, basically. So this is another way, like alternative promoters are one pretty cool way of generating different proteins from the same gene. Alternative splicing is another one. And so I think that's actually pretty funky. This is not like super well understood, because it's, yeah, you have to figure out what different mRNAs are being produced from a gene to be able to figure out how those are being generated, essentially. Okay, I think we have enough time for RNA stability. Probably not enough time for post-translation modification. We can do that in the second class. Okay, so essentially what we've done so far in all the steps from chromatin through the control of transcription, the control of RNA processing, assumes that those mRNAs are equal. That they're all going to be the same in terms of translation. And that's not the case. So there is a vast range in terms of how long those mRNAs hang around for. And basically, the longer an mRNA persists for, equals the more protein produced using that mRNA. So we have two kind of deals going on here. So A, how much protein is produced, and B, how long is that protein produced for? Both of which tie into how long that mRNA hangs around for. And so if you want to produce only a small amount of protein, either you transcribe it, the gene at a low level, or you only allow that mRNA to hang around for a short amount of time. Right, so that kind of gives you I don't know, a better control if you think about the protein, right? So if you need more protein, then you just express the gene for a little bit more, right? It's a little bit, gives you a little bit more control over protein quantity. The second aspect is, well, you know, sometimes you don't want genes to be expressed forever, right? They have a specific kind of time window in which they are needed, after which they may do more harm by hanging around. So essentially, if you can't have any way of getting rid of mRNA as soon as you turn a gene on, if that mRNA hangs around forever, you'll keep getting protein made forever, because as long as that mRNA is around, it's going to be translated by, you know, translation machinery. Sorry, my dog is twitching, so it's kind of distracting out the corner of my eye. And so if you want to shut off a gene, right, you want it for a very specific kind of time window, maybe specific point in development or specific response to a stimulus, right? Ingest a toxin, express the genes responsible for detoxifying that toxin. Once you don't have any toxin anymore, you don't need those proteins being produced anymore. So to turn off gene expression, not only do you have to stop transcribing the mRNA, you also have to get rid of the mRNA that has been made already. And the way we do that is by essentially degrading that mRNA. Right, so remember way back when we talked about protecting the ends of an mRNA, 5' cap, poly A tail, right? We need to kind of expose the inside of that mRNA to RNases, right? And we do that via a couple of different mechanisms, involving what's called RNA silencing. So there are two aspects to RNA silencing. One is RNA degradation. Second is inhibition of translation. And so essentially, you have two different forms too. MicroRNAs inhibit expression of endogenous genes, in other words, your own. Small interfering RNAs, or siRNAs, inhibit expression of viral and, well, mostly viral genes. Right, so you have two kind of different things. And again, microRNAs and siRNAs are non-coding RNAs. Right, so they don't encode a protein. Their job is not involved in like the normal path of gene expression, producing proteins. Their job is to target mRNAs for degradation or inhibition. And actually, we'll probably need to continue this in the next class. Yeah, I think that's probably a good place to stop, to be honest. So have a look through this in the break if you have time, and we'll come back at 12:30. Because I don't really have time to properly go through this in the last 2 minutes. I don't want to rush it. Okay, so see you in 17 minutes. Stretch your legs, have a wander around, and we'll finish off eukaryotic gene expression when we come back, and we'll have a look at some cool applications of what we've learned. All right, so see you at 12:30 and have a nice break.